In today's video, we're going to be covering ATP and inorganic ions. We'll start first with ATP. ATP is a source of energy that is used by cells within an organism. So animals and plants use glucose as a big source of their energy. So in animals, we will take in glucose from our diet, and in plants, they will make their own glucose and their own sugars through photosynthesis. And energy is released from glucose in the respiration process. And this is because cells aren't able to use energy directly from glucose. So respiration will release energy in a form that is more usable by the cells. And in this case, it releases ATP, which stands for adenosine triphosphate. So in the left-hand corner of the screen, we have a molecule of ATP. And the structures of this molecule should start to be very familiar to us now. So we can see we've got a ribose sugar, we've got an adenine base. So this base and this sugar together make a molecule known as adenosine. And we've got 3 phosphate groups, hence the name adenosine triphosphate. Now in your exam, ATP is a perfectly acceptable abbreviation to use. Your examiner will know what you're talking about. ATP is what's known as a nucleotide derivative, and this is just because it's a modified form of a nucleotide. So if we think back to what a nucleotide looks like, we know that it has a pentose sugar, and in ATP we have a ribose sugar. We know that nucleotides always have a nitrogenous base. And in the case of ATP, we always have an adenine base. And we know also that in nucleotides, we will have a phosphate group. Now, this is where the modified nucleotide form comes in because ATP has 3 phosphate groups. So when a cell requires energy, ATP will diffuse to that part of the cell, and then the bonds between these phosphates will be broken by a hydrolysis reaction, and that is where the energy is released. Let's now have a look at ATP synthesis and breakdown. When a cell needs energy, ATP will diffuse to the part of the cell that requires the energy, and it will be broken down into ADP, which is adenosine diphosphate, meaning it only has 2 phosphate groups, and PI, which is an inorganic phosphate. So this is our 3rd phosphate, the one that is cleaved off, that goes on to form an inorganic phosphate. Phosphate bonds are broken by the hydrolysis reaction. So water will come in and it will cleave the bond that holds these phosphate groups together. And this is catalyzed by an enzyme called ATP hydrolase. So if you're struggling to remember this, we know that many enzyme names end with A's: catalase, maltase, nuclease. So we know that A's will be on the end, and this is an enzyme that is involved in ATP hydrolysis, so therefore the name is ATP hydrolase. And when these bonds are broken, our energy is released. And ATP hydrolysis is coupled to other reactions in the cell that will require energy. So an example of this is active transport. So ATP breakdown will provide energy needed for active transport to occur. And this just means that the energy from ATP is used directly when reactions are coupled and minimizes the loss of energy as heat. So it just increases the cell's energy efficiency. The inorganic phosphates that are released are used in a process called phosphorylation, which is where a phosphate group sticks onto a compound and makes it more reactive. ATP is resynthesized by a condensation reaction between ADP and PI, and this is catalyzed by ATP synthase. So if we think about the enzyme name, we know it's an enzyme, it's going to end in A's, and it's to do with ATP synthesis, hence ATP synthase. So now let's have a look at the conversion process between ATP and ADP. On our right hand side of the screen, we've got an ATP molecule, and we know this because there are 3 phosphates present. And then on our left, we've got our ADP, because there are only 2 phosphates, and this purple dot that's floating around with a PI in the centre is our inorganic phosphate. So to go from ATP to ADP, we know that a hydrolysis reaction is going to occur and energy will be released, and this is catalysed by ATP hydrolase. And then to convert ADP back to ATP, we know that our ADP and our PI are going to need to bond together again in a condensation reaction, and this requires energy. So for ATP synthesis to occur, energy is going to be needed. And this activation energy is lowered by ATP synthase. Now, the reason that this reaction requires energy is because if you think about it, if energy is stored in the bonds between phosphates, when those phosphates join together again and that bond is formed, we need energy to go into that bond. So that about covers it for ATP for now. It's really important that you get to grips with these facts, because in the A2 syllabus, one of the first topics we cover is aerobic respiration and anaerobic respiration, and we will be looking at ATP in a lot more detail there. So moving on to inorganic ions now. An ion is an atom or a group of atoms with an electric charge, and this can be either a positive or a negative charge. So we can see by my examples, we've got Na+, which is a sodium atom. Na represents the chemical symbol for sodium. We can see it's got a positive charge. Now, with atoms that have or groups of atoms that have a charge of only +1, we just write the plus symbol or the minus symbol if it's got a negative charge. It's only when the ion or the has a charge greater than 1 that we write a number. So if it's got a charge of +1 or -1, we just write the symbol. So for phosphate, we can see this is a group of atoms now with an electric charge. It's got a charge of 3-, so we write the number of charge, which in this case is 3, and we write obviously the symbol to show that it has a negative charge and not a positive charge. So ions with positive charges are called cations, so sodium is a cation, and ions with negative charge are called anions, so in this case phosphate is our anion. Inorganic ions are ions that typically don't contain carbon. There are some exceptions, and the one I found is carbonate, which is CO3 2-. However, I don't believe this is covered in A-level biology. I think it might possibly be touched upon in A-level chemistry, but it's not one that you'd really need to know about for this course. Inorganic ions will exist in solution either in the cell's cytoplasm or in bodily fluids. So, for example, there might be sodium ions within the urine, or there might be potassium ions within the cell cytoplasm. And ions all have specific roles depending on its properties, and the role that it has will determine its concentration in certain environments. So if you think about cells now, when cells become, or neuronal cells, when these become excitable, there is potassium ions that leak out from the inside of the cell to the outside of the cell, causing this cell to become depolarized. But when that cell is at rest, the properties of the potassium ion in the process of depolarization mean that when the cell is resting, there is a really high concentration of potassium within the cells compared to outside the cells. Now let's have a look at the function of some of the ions that we will need to know about for this course. We'll start off with hydrogen ions, which have the chemical symbol H+, and hydrogen ions determine the pH of a solution. So if there are more H plus ions present within an environment, the pH will be lower and the environment will be more acidic. So enzyme-controlled reactions are really affected by changes in pH. So we know that our enzymes all have an optimum pH, and if there are suddenly more hydrogen ions present within the environment that this enzyme-controlled reaction is occurring in, these hydrogen ions will cause the bonds in our enzyme to change and be broken, and this will cause a change to the enzyme's active site, meaning that our reaction rate will first slow down and then it will stop completely. Another really interesting example of an environment that needs to have the pH carefully controlled is within the blood. So the blood needs to have a pH between 7.35 and 7.45. And this is really important because if the pH of the blood becomes too low, the blood gets more acidic, you enter a state of acidosis. And if the pH becomes too high and goes above 7.45, you enter a state of alkalosis. And this can have really serious effects on your health. Our next ion that we're going to look at are iron ions, and these are a really important part of a molecule called hemoglobin. And hemoglobin is a protein that carries oxygen around the body via the red blood cells. Hemoglobin is made up of 4 polypeptide chains, so we already know that it's a quaternary structured protein, and it's made up of 2 alpha and 2 beta chains. Now, if you imagine a square, there is a chain, a hemoglobin chain, at each corner of the square, and in the centre of the square, or in the centre of our hemoglobin molecule, we have an Fe2 plus ion. Now, iron is what's known as a transition metal, which means it can change oxidation states. So when oxygen binds to hemoglobin, our iron can transition from an Fe2 plus ion to an Fe3 plus ion. And when oxygen is released at the site where it is needed, our Fe3 plus transitions back to Fe2 plus. Our next ion that we're going to look at are the phosphate ions, which we saw earlier, PO43-, and these can attach to other molecules and phosphorylate them, and when it attaches to other molecules, it's known as a phosphate group. Bonds between phosphate groups can store energy, like we saw in ATP, and phosphate groups are very important in DNA and RNA because they cause that bonding which converts them from one single nucleotide into a polynucleotide chain. So if we think about a nucleotide, we know that we've got our phosphate group sticking out, and this phosphate group will bind to the pentose sugar in an adjacent nucleotide, forming that phosphodiester bond. And our final ion that we're going to look at is sodium, or Na+, and this is used in the co-transport of molecules across cell membranes. So for slightly larger molecules, like glucose or amino acids, these molecules can't cross the cell membrane by diffusion through the membrane. They have to be transported into the cell via a receptor on the cell surface membrane. So glucose or an amino acid, it will come along, it will bind to its relevant receptor, but it will also need a sodium ion to bind to this receptor as well, so that it can either enter or leave the cell. So let's recap everything that we've covered today. We know that cells release energy from glucose in the form of ATP, which is this form that the cell can use. ATP stores energy between the phosphate groups. We know that ATP is broken down into ADP and PI by ATP hydrolase in a hydrolysis reaction, and conversely, ADP and PI can join to make ATP in a condensation reaction, and this is catalyzed by ATP synthase. We know that ions are atoms or groups of atoms with an electric charge, and that inorganic ions don't contain carbon, and they can include Na+, for co-transport, H+, which is used in the control of pH, Fe2+, which is important in hemoglobin, and PO43- or phosphate, which is found on DNA, RNA, and ATP. So now that we have covered this topic, it's time to test yourself with the following questions. Number 1, draw the structure of an ATP molecule and label its components. How is energy released from ATP? Talk about the reaction type, the bonds that are broken, and the products that are formed. How is energy loss through heat minimized when ATP breakdown occurs? What is an ion? What is the difference between organic and inorganic ions? And finally, name 3 types of inorganic ion and describe their function. That's all for today. Thank you for listening.